Hi, I'm Deborah Schmidt. I'm a research software engineer at the Max Delbrück Center in Berlin in the Image Data Analysis Group. And I'm here today to present what we have worked on for Helmholtz imaging and for the whole Helmholtz Association to improve reproducible software handling for scientific solutions. The tool I'm about to present, which is called Album, is developed by Philipp, Albrecht, Harrington, and myself. Our group provides support for Helmholtz imaging, which is an incubator platform of the Helmholtz Association. We mainly support the excellent science done by Helmholtz scientists all over Germany, but we are also able to support you if you are working on science or if you work in industry. We provide project funding for cross-research areas, and there's a help desk that you can just mail to in order to get in contact. Today, we are tackling a problem that we have seen often in the past when working together with biologists, for example. They have very specific problems that need to be solved computationally, and they know that there are many tools available that could potentially solve their problem. But it's just really hard to figure out how to install them, how to use them, or to be able to reproduce something that someone else did for a publication. With Album, we have this vision of scientific research groups being able to share catalogs of specific solutions describing a scientific problem and how they dealt with it. And these solutions can then link to specific tools. The framework that we wrote to support this vision is called Album, and it's a decentralized distribution platform for solutions to specific scientific problems. The command line tool is already published and the graphical user interface exists as a prototype. You can find the preprint and all the documentation on our website at album.solutions. Album solutions are fair. You might have heard of these principles also in relation to scientific data management. These solutions are findable. You can attach rich metadata. They are accessible. We have tested the whole framework to work on Linux, Windows, and Mac OS. And we are also working on exporting things into common formats, like, for example, Docker. We are putting much effort into giving best practices on how to achieve reproducible solutions. Each solution has its own environment, its own Conda environment with fixed versioning, and everything is open source. In which situations could Album save you time? Well, for example, if you want to create reproducible routines, not only for publications, but also for yourself in 6 months, or for a colleague that's just starting new. Then there might be computational tasks that you do every day where you have to do a couple of clicks here and there that we could translate into a solution that could automatically call this. The catalogs that you can build with Album could highlight specific use cases for existing software packages. So we still want everyone to use the established ways of publishing and deploying software packages like PyPy or Maven or Conda, but with Album, we could describe how a specific software with specific parameters and a specific plugin can be used in a scientific case. Now let's look at what the solution actually is. A solution is a single Python file. This Python file includes a setup method, which provides the group name and version of the solution, that's the identifier, and then the API version that you're using for the specific solution. A findable solution has metadata attached to it, and you can provide something like a title, descriptions, authors, citations, and we try to stay as close to the bioimage.io format here, which is another great effort of making software across multiple tools useful for deep learning and server-based approaches. Now this solution has all the metadata, but it still doesn't do anything. So let's look at a solution that is actually executable. This solution here provides a run method, and this run method is also a Python method that can literally do anything, and you can provide arguments which you can access in the run method. is not everything that you can do with the solution. You can describe a specific Conda environment, you can provide custom install and uninstall methods, you can provide test methods, and much more, and all of this is documented on our website. What is a catalog? A catalog is a Git repository, and this Git repository could live on a hard drive somewhere, on a network drive, on GitHub, on GitLab, all of this works. If you deploy your catalog to GitLab, we already have scripts set up to generate a web page for the catalog automatically, which means that you can browse the available solutions online. That's what you see here. And if you click on a specific solution, then you see here the tags and the citations and the arguments that you can provide for the solution. Now we use this for Helmholtz imaging, where we deal with images from many different domains. Each domain has their own tools, and we want to make it easy to access these tools also if you're not used to them. So we have a project where we want to use LabKit, which is usually installed through Fiji and as an update site. And we wrote a solution to just launch LabKit directly with an image and be able to segment and use the random forest classification in LabKit in the easiest and direct way as possible. Here's another example. That's a project I was involved in my previous job at MPI-CBG. It's led by Andreas Müller, and it's a three-dimensional reconstruction of a beta cell and all of these organelles in the beta cell, and used very different approaches to segment all these different organelles. We also put much effort into the plots, but since this is all so complex, it doesn't make it easily reproducible to just publish the code. Therefore, we will continue this collaboration and work on solutions for each of these individual steps. And this is how it could look like. It could mean that there's a solution for downloading the data, a solution that helps you to sparsely label your data and then do the different deep learning training and prediction steps in Elastic, but also, for example, with Stardust and Python. Then there can be solutions for the data analysis in ImageJ and for the mesh generation that we did in SciView in this case, and then the plot generation in Python and the final rendering in Blender. So we're looking very much forward to share these solutions with you in the future. What you see here, that this is quite a complex procedure and that these solutions need to work together. So we are also exploring how to be able to integrate them into some kind of workflow. We don't want to reinvent the wheel. That's why we want to use existing frameworks for this. And in order to do so, we published the Python API for Album. There's also REST API, and we will work on best use cases for how to streamline solutions into single solution workflows. In order to show a prototype of this, I'm displaying the graphical user interface of Album here, which we will release soon. This is just something that lists your local collection and all the solutions that you already have available through the catalogs that you added. And then this one solution that we are launching here is accessing multiple applications through other solutions. In this case, first, it's using VTK to generate this mesh. That's the Helmholtz imaging logo. Then the mesh is stored. It is loaded again with Blender. Blender is now rendering this mesh. And all of this is happening automatically. But as you can see, the graphical user interface is opening, and we could also add options here to do manual adjustments in between, which is pretty neat. At the end here, what we see is that the rendering is stored and then opens in Apari just because we can. The plan of our group for this year is to focus on collaborations and on establishing the Helmholtz Imaging Solutions catalog. That's it. Thank you for your attention. You can contact me ideally via the help desk at helmholtzimaging.de address. Then you can ask for our group or for me or for album support. There's our website again with all the information. I want to thank everyone from our group, especially also Jutta Steinkötte and Dagmar Keinmüller, who are supporting us after Kai left to Oak Ridge National Lab, our PhD students Leo and Madhu, and also Lukas Riekert, who is new to our team and supporting us with the software development. Thank you.